Hi students, so today I am going to start with the process of translation. So translation means the passing the information or the messages from mRNA to proteins, and the mRNA is the messenger RNA which is carrying the information from DNA and that will be passed on to the proteins. So in this video today we are going to discuss about the requirements. What are the requirements for translation? So before we start with that, let's have a revision of the definition of translation. So translation is the process of polymerization of amino acids to form the polypeptide. When the amino acids they combine to make a polypeptide or a protein chain, it is called as translation. The order and the sequence of amino acids are defined by the sequence of bases in the messenger RNA. Whatever is the sequence present on mRNA, accordingly the different amino acids will be packed, and then finally the amino acids are joined by the peptide bond. So we can say that joining of the amino acids with the peptide bond to make a polypeptide chain according to the sequence present on messenger RNA is called as the translation. So we'll discuss about the requirements for translation in this video. So the requirements are important requirements are the messenger RNA, ribosomes, and the transfer or the tRNA. So let's start with the M or the messenger RNA. As the name is explaining, it is a messenger RNA. That means a segment of DNA from where the message has been carried by a RNA. So you can see this is the messenger RNA. Messenger RNA, for the detailed structure you can go to my video, that is the transcription in case of the eukaryotes. So let's discuss its main parts. It is having this green segment which is called as the coding sequence. Fine. This coding sequence is the sequence which is carrying the messages from DNA, and according to the code present on this coding sequence, the different amino acids will be formed. So here is one typing error, sequence के spellings में थोड़ी mistake हो गई है, please check that. Fine. So this coding sequence is a sequence present on messenger RNA which carry the message from the DNA. Then it is having a methylated guanine cap at 5' end. There is a polyadenylated tail at 3' end, and these two yellow colored segments shown over here, they are called as the UTRs. Very important question, what are UTRs? UTRs are the untranslated regions which do not translate. It means this segment and this segment of messenger RNA do not code for any amino acid. So this is the structure of messenger RNA. Then the next one is the ribosome. This is an important component because they are called as the protein factories of the cell. So these protein factories are having two subunits, smaller and the larger subunits. These units are present freely. When they have to synthesize the proteins, these two subunits, they will come and they will join together. So for joining them, first of all the smaller subunit will attach with the messenger RNA, and then after that, after attaching with the messenger RNA, it will attach with the larger subunit. So you can see here, this is the mRNA binding site present on the smaller subunit, and the larger subunit is having three important sites, A, P and E. Fine. A is the acceptor site which will accept or which will receive the tRNA carrying the amino acid. P is the polypeptide site, and where the chain of polypeptide protein will be formed, and E is the exit site. The detailed step will be discussed in the next video where we will be making the proteins. So these all the three points will be more clear in that video to you people. And an important thing is there, the two subunits of the ribosomes, they attach in the presence of the Mg2 positive ions. Then the next important component for the protein synthesis is tRNA. tRNA is the transfer RNA which has to transfer the amino acids from the cytoplasm to the ribosomes. So if we talk about the structure of tRNA, you can see this is the two-dimensional structure, this is the three-dimensional structure. In your previous editions of NCERT, this structure is there, and in the latest edition, this structure is there. So in case of the that your tRNA, now the important part is One is the amino acid binding site. You can see this is the amino acid binding site at the 3' end where the respective amino acid will attach. So you can see the similar amino acid binding site is present over there also. Then this is the anticodon site. This anticodon site is having the sequence which is complementary to the sequence present on messenger RNA. You can see this is the code present. This is mRNA strand, code present on mRNA GAG, and this is the sequence present on the your tRNA, that is CUG, complementary to the GAG present on the mRNA. Fine. So this is the anticodon site. Same over here, anticodon site. Even sometimes the chain of the tRNA, they can be self-ligated, so they will be having intramolecular base pairing. Then important one is there, the attachment of the amino acid at the 3' end. So this is all about the requirements for the process of transcription. The process of transcription I am going to discuss in my next video. Thank you.